would you mind introducing yourselves?
Sure. My name is Kostis, Karagiannis. I work for Moderna. I'm the director of the bioinformatics engineering, focusing on the individualized neoantigen therapy. We were using a lot of Nextflow for this and other pipelines in Moderna. In my past life, I've been the lead bioinformatics scientist of one of the centers of the FDA, where Nextflow was also part of the work there.
And it's great to see everyone again, and thanks to the organizers for the great conference so far. But I'm Brice, for those of you I haven't met. I am in ZS. If you're not familiar with ZS, it's a management consulting firm. We have about 13,000 people worldwide in a number of different offices. But I'm specifically part of our ZS Discovery organization, which focuses on answering scientific and technological questions kind of all across the R&D life cycle. There, I have responsibility for being the lead of our bioinformatics track of our Medical and Scientific Expertise Center, as well as the lead for a solution we have focused on developing and deploying advanced bioinformatics workflows, so lots of Nextflow, which is why you see our presence here at this conference and over in Barcelona. Prior to this, I was leading computational genomics at Johnson & Johnson and spent some time in biotech and in academia prior to that.
Cool, thank you. So just a bit of audience participation. Who here doesn't know what GXP is? Okay, cool. So I guess that's a good segue for an introduction to a quick overview of what GXP actually is.
Sure, I can get started. Well, maybe also a relevant question would be how many people have worked in a GXP environment, right? Let's see the hands on that. Okay, so that gives me a little bit of a perspective of how to present it, right? There are many ways to approach it, but having been involved in GXP systems and in my past life not being involved in GXP systems and purely doing academic work, in essence, in a very quick pitch, you could say that it's a lot of things that you already do, that you already want to do and follow these good practices. They're all these good practices that people have in their mind, formalized with specific documentation, with specific processes, that those documentations and processes will be eventually one day, or is expected that eventually one day will be audited. And that's what makes it GXP. So these are the good practices that will ensure that the product that is being used or being developed by these processes will be meeting the quality as it's been described by those processes. That's a very broad definition. We want to go to more details.
Yeah, maybe just to provide a little bit more flavor. For those of you who aren't as familiar with the terminology, just as the G, the lowercase x, and P, so good something processes, treated as a variable, good laboratory processes, good manufacturing processes. And when we think about this from a bioinformatics or computational situation, what we're really considering is a variety of different types of layers of things that need to have all the documentation that Kostis just mentioned here. So the infrastructure itself that's running on needs to have documentation that shows it's doing what it should be doing. Same with the platform that you're running your analysis on, whether that's Sakara or a variety of other types of platforms. And same with your bioinformatics workflows. It needs to be producing appropriate results. And if anybody here in the room has worked in genetic testing or under kind of CLIA environments or things like that, you might have come across this in a little bit where if you, say, change a program or want to update something, even just a version of a tool, or try out something new and subbing it into that, you need to show that the results that are coming out are consistent with what you have previously. So there's all of this more formalized process that's baked into it. And again, at the end of the day, the whole point is if it comes to a point where you're audited for a variety of different reasons, maybe by the FDA, maybe by other organizations, that you can show that, yes, we've kind of dotted our I's and crosses our T's to get to the point where we have all that information that's available that can show that things are consistent.
Cool. Why is this relevant to Nextflow and the audience here?
Yeah, so I'm a scientist too. I did a PhD in working workflow languages for a while, so like many of you here. And I also spent time in pharma. And what's really interesting is that in early research, when we're trying out the hot new tools, trying out the hot new data sets to analyze, we can poke around and pretty much do whatever we want in terms of how we're building things out. Code doesn't need to be standardized. I think we've all dealt with refactoring types of engagements. And have worked in environments that are not as nice as what Isha just showed in the previous conversation. But if you want to take that and it needs to be producing results that have direct impacts to patients in a variety of different types of ways, either in terms of the way that the drugs are produced, that you're appropriately capturing things like maybe different toxicity patterns, are the drugs being targeted for the right populations, all of the core scientific questions that would go into pharmaceutical R&D. This type of process needs to be kind of baked in so that the results that are coming out are ultimately reliable, so that the drugs that are produced are both safe and efficacious for patients. So from a Nextflow framework here, we can of course develop our workflows to do ultimately whatever we need to do to explore our science, but that formalized process needs to have a level of rigor beyond just putting it into production at the end of the day to make sure that it stands up to that extreme high level of scrutiny when you transition from research and development into the clinic.
Yeah, very well said. And actually, I can take it one step further, which is what excites me in the space in general, right? Being on both sides of the industry, regulated and regulatory, bioinformatics has been traditionally considered as the stage of research. So there are a lot of products out there that their life cycle started from at some point from a bioinformatics pipeline. You know, you do the biomarkers, you're trying to find what's the thing that you want to target, whether that's a gene or anything, but that's the research stage, right? And you need all these good practices for this stage, as Brice mentioned, right? That's something that you really want to be focusing on. But things change dramatically once you start looking at precision medicine. Now your bioinformatics pipeline is no longer the thing that happened in research. No, it's very much part of the treatment, right? There's a big conversation where do you classify it? Is it like manufacturing? Is it something else? This is not the point of the conversation, though. The point is that the Nextflow, your pipeline, is the very thing that needs to be part of the process that has been traditionally in a GXP system. So you need to be part of what is having a very strong provenance of information, right? And Nextflow has been doing a lot of work towards that. There has been a great initiative for BCO, for instance. I'm connecting both words here. BCO is a framework that started from FDA on how to capture the provenance of your bioinformatics pipeline and in a way to communicate with your regulatory authorities. What you've done, exactly with what data, exactly with what purpose. That's very important. You know, you can take a DNA pipeline, a somatic pipeline, and run it as a germline. Sure, what does it mean? The error domain, and a lot of other things. And Nextflow allows you to create that BCO content that would be easily transferable to your regulators. Not only BCO, right? I don't want to monopolize that concept. There's also RO-Crates, which serves a very similar purpose. So the very notion that now bioinformatics becomes part of the product that it's treating patients, that kind of like increases bar for how we meet the GXP requirements.
Yeah, that's a good point. Nextflow is quite key for the turning the handle on the analysis and that sort of stuff, but there is a whole bunch of other thing around that needs to be validated and, you know, documented and passed on to the authorities. I also know there's community initiatives around Nextflow in NF Core, for example. There's a regulatory SIG. Do you want to maybe comment on that, Brice?
Yeah, I think, you know, we're right at the forefront of this as the people in this room and as a field generally. So there's a regulatory special interest group that is working together. There's a number of partners from different pharmaceutical organizations. And the question is really, when we think about putting together this documentation for bioinformatics, especially with respect to validation of bioinformatics pipelines, what does that look like? There's not something we can go to right now and say, oh, this is exactly what we need to do. So just like how pharmaceutical companies often engage with the FDAs in various ways to go through and say, hey, you know, we have this new technology, we think it should be used as evidence for in our, you know, IND filings for first-in-human studies or things like that. We have the opportunity as a community to basically come out and say, this is what we think the appropriate standard should be, and let's figure this out as a group moving forward. So it's a very community-based activity, and the special interest group is one way to tackle that.
So there's, in general, the whole process, there's lots of different things that you need to tackle on different fronts. You know, there's the IQ, OQ, PQ buzzwords going across all the way to try and, you know, end up with, ideally with a validated environment. does it all have to be tackled in serial, or is there a way that you can chunk it up, do things in parallel, optimize time to production, clinical sort of pipelines and analysis and that sort of stuff? How would you tackle that kind of thing?
Yeah, that's a very good question. It's kind of like diving a little bit on what we've been learning as well in the process. I'm not sure if it is a true statement, but when I started that work in the bioinformatics and GXP for products that they can be part of for processes that can be part of a product, I got a feeling that this is kind of a first attempt. We didn't have a guidebook on how things are done, so there was a lot of room for improvement on how the process works. As you mentioned, like the validation is a very specific term, right? I don't think that we need to go into these details, but there are certain steps. You need to do your installation qualification, practically make sure that you deploy your pipeline as you're expected. You're testing your pipeline, right? But there are other documents as well, like your design document. So these are very well-defined steps in the process, but these very definitions are coming from a paradigm which I don't know if everybody knows here, the waterfall practically development, which I don't think that people are as familiar as with agile these days, right? So to map something that we've been traditionally doing with bioinformatics, and it's more on an agile stage, to a framework that's Nextflow, we had to reinvent a little bit the processes. So there is some CDS that it's happening, meaning that we're defining the specs while we're creating the OQs, and we are creating this parallelization as much as possible. So we're trying not to stick to the time points, at least in terms of creating the drafts and the documents. Approvals and everything else is it's a different conversation.
Yeah, I guess one thing I'd mention here is that if you do this process, and those three kind of layers that I mentioned previously, they can be done, once, and talking the infrastructure at one point, the platform in another part. And yes, everything's kind of connected, but there's an opportunity to do some things in parallel. If you do it once and you don't make any changes to it, you don't need to keep doing that day in, day out. But if you do make a change to the process, that's when you have to go back and basically reassess and show that everything is working the way that it should be. So that's going to be most frequent, I would argue, at the level of the bioinformatics pipelines. New tools, changes, a testing process that's baked in, showing that the results come out in an appropriate way. But if you're using, for example, the Sakara platform and there's been no updates pushed or anything to that, you don't have to go through this process. So it's kind of a one and done in some layers unless something else happens.
Given your experience with the process, sort of lessons learned, how can you improve this process? How can you increase turnaround times? How can we become more efficient with, getting to that end state where you are GXP validated?
So maybe I'll answer this in a very high level way with a real world example, and then Kostis, maybe you can jump into. Kind of a lighter weight way of putting this, it might resonate with this room. We've engaged with clients where the R&D IT organizations will have potentially a GXP environment. Say, the IDEs, standard platforms that folks would work with. And it's actually quite challenging for scientists to come in and work in that. You can't test things out, you can't try the new version of tools, you can't update system libraries, you can't add additional resources because of everything that was put into that and kind of the months worth of work that went into that process. So there's an opportunity here from a broader conceptual level, I think, to, as scientists, engage with R&D IT organization, make sure that the scientists understand why the regulatory pieces and the lockdown in the process is important, but then that IT organizations realize that, hey, we're scientists. Things need to be flexible. We need to have the opportunity to poke around with data and analyze it the way that we want and test out that hot new thing that came out in Nature last week. And the correct answer for kind of day-to-day work is somewhere in the middle. So there's nothing wrong with having a GXP and a non-GXP type of environment. If you go only one or the other direction, then you end up in this weird middle area where you're not necessarily solving the problems of both sides of that. So maybe for this audience, I'll leave it there. But
yeah, no, I echo that. In our experience, I think what has enabled us to move a little bit faster than expected, I'll be honest here, is the cross-training. We had to train both sides. As Brice very well mentioned, we had to train our bioinformaticians to understand that things cannot be changing as easily as Nextflow makes it, which is great. It's a great tool indeed, but it's not possible to do it without the processes in place. But also train our QA and other functions and regulatory functions to understand that it's impossible to develop in a waterfall model where you deploy something and then you forget about it. That's not how bioinformatics works. That's not because it's a huge scientific effort, and you know that there are going to be improvements constantly, and you want to bring these improvements to the patients, right? That's the whole point. So cross-training was a major part for our process. And what I also realized that it was tremendously helpful is to try to give a little bit of the taste of the to the bioinformaticians and all the stakeholders involved of the auditing process. So practically, when you have a big team, as Isha was mentioning, right, working on the same pipeline, if all the members in the team are awfully familiar with all the aspects of your pipeline, then it's very hard to understand what somebody outside of the picture will be asking. So trying to create some silos on purpose inside the team help to remove ourselves from the picture and have that a little bit of an auditory hat, if you may. That was a small experiment, just that seems that it has worked so far. I don't know, it's a brand new space, and we're trying new ideas.
Cool. So we've got a couple of minutes. I can actually, in fact, 1 minute for a question. There was a question here from Olga about, I don't know if you're at liberty to say, I know you're going through the process right now, but do you have a specific example of something biologically related in terms of a pipeline that's in Nextflow that you're using as a validation for this?
Well, I would point back to my title in the individualized neoantigen therapies. I think that it makes sense that all the precision medicine-related pipelines that could be considered as part of a system that needs to be GXP, right? Now, to what extent this is a common practice in the industry, I don't have a view on that, but I believe it's a reasonable approach.
One example that we see that might resonate with folks in this room is cell therapy stuff. So it's in cell therapy, the product that comes out, the manufacturing process is the therapy. And yet there's genomic interrogation of those cells to make sure that everything's expected before you put it back into the patient. You know, collect their blood, do some stuff, put it back in the patient. That needs to follow a good manufacturing and good laboratory process, and the analysis needs to follow, you know, the same types of things we've been discussing today as kind of software as a medical device. So that's the one I think is very tangible, and people that have produced those types of therapeutics recently, is, you know, and they're very successful, have needed to basically interrogate their bioinformatics and their manufacturing that way as well.
Great. I think we're just on time. Thank you for your time. And